right, everyone. Thanks so much for coming. This course is kind of intended to be everything that I've learned with fNIRS so far in our lab has been mostly self-taught, and the point of this is to like dump all of that into you. So if I'm like hit by lightning tomorrow, the lab doesn't lose anything. I'm going to be recording these slides in audio. I'll be posting those on the lab drive. If you guys have access to the SCN members lab account, it'll be just in a drive folder called like I think fNIRS Bootcamp 2019. And if you don't have access to that, I will send files and stuff to appropriate people. Just let me know if that is you and you need stuff sent directly to you. Okay, also code. There'll be code on Friday. Today's and tomorrow are going to be mostly stuff that's lectured at you. Today is going to be kind of like an fNIRS intro. Most people here have used it to some capacity or know about it, but for those who don't, there'll be kind of an easing into it, as well as some physics about fNIRS that you're not going to need to know by heart through what you do, but like you'll probably want to be familiar with it, and so that you can write a method section. And when people tell you like, oh yeah, Beer-Lambert Law, ooh, you know what they're talking about. And then tomorrow is more into the first part of like collecting data, doing that well, pre-processing data, and then Friday is all about analysis, data visualization, and just playing around with data. There'll be sets that you can download that we can all work on together. So for today and tomorrow, just have some capacity to take notes if you want to take notes. For day 3, make sure you have a laptop that can either run MATLAB 2014b, I believe, or later, or Python, because I'm going to try to make sure all the analysis code we have is going to be written in both MATLAB and Python versions for whichever language you prefer. Okay, so for today, I'm going to again do what is fNIRS, look into a brief intro and how it compares to other modalities that those of us in the room may be familiar with, why you might want to choose fNIRS over others, some history about the imaging technology so you know where it comes from, the physics lesson, and the first part of high-level overview of what you need to be thinking about when you're designing an fNIRS study. So what is this thing? Well, it lives in that room. And fNIRS stands for Functional Near-Infrared Spectroscopy. It's a non-invasive neuroimaging device and kind of akin to a pulse oximeter at the doctor's office. That's just using one little near-infrared emitter and detector to measure oxygenation in your blood flow in your finger. We're bringing that out to looking at blood flow over the entire head. Functional stands for you're taking lots of pictures of the brain over time to identify changes in activity. And it relies on the optical properties of cerebral blood flow to infer brain activity. We're going to go more into this a little bit later, versus like MRI that we're really familiar with in this lab, which is using the magnetic properties of cerebral blood flow instead. The machine itself, we're using a specific machine from a specific distributor, so that's what this one looks like. Some other machines will look a bit different, but they're generally a smallish box between a textbook and a microwave in size. By the way, I'm going to have lots of text on these slides so that you can like refer to them later, so don't worry about like feeling the need to read through this and listen to me, because I'm mostly just reading through this. And our machine at least connects to a computer via USB. It includes bundles of what's called optodes. These are the wires that have the optical components through which you're doing the signal detection analysis. And these optodes, which are hanging at the end of the wire there in the picture, are typically held to someone's head via like a stretchy cap that we use, or it might be inserted into some sort of rigid frame which is then strapped to someone's head. When we talk about fNIRS and fNIRS data, we often say channels. A channel is the data recorded between one source and one detector pair. And that's detecting activity in the brain area that lies between those two optodes. The number of channels possible is determined by all the unique pairs of sources and detectors that you have on someone's head. Our machine specifically is a NIRS Scout unit from the company NIRS. It has 32 sources and 32 detectors. We also just got a smaller unit called a NIRS Sport that can be run wirelessly, that has 16 sources and 16 detectors. Other popular brands, just for your future reference, are Hitachi, Shimadzu, Artinis, Biopac, and Tekken.
I have a question.
Yeah.
Do you know how popular each of these are?
So how popular each of these are, I probably listed them roughly in order of popularity, actually. A lot of people enjoy the Shimadzu and Hitachi units, especially if they're running labs that are in Asia. There's a lot of NIRS labs in Japan and Korea and China. Artinis is the unit that's probably the most bang for your buck. It's a cheap unit, but it's good for when you don't have like full head coverage that you need and you want to have something that's not like upwards of $100,000. There are also much smaller companies, but I don't really see people talk about them, so I don't think you really need to know them. Why we use it, the big question is, why does this exist in our lab? It's cheaper than MRI, but we've still spent a lot of money on it, so what's the purpose of this? We generally want to strike a nice sweet spot between really careful, detailed brain activity recording that you can get really well with MRI, but that with MRI you can't quite do rich naturalistic experimental paradigms very easily, especially anything where a participant is actually embedded in a real scenario. MRI can only kind of get them to approximate by watching a screen of something. So fNIRS can get us in that little between area more easily than other neuroimaging methods can. It's also, relative to our interests, it's more affordable to run participants than through fMRI. fMRI, you have kind of scanner fees that you have to pay to the center each time you do it, and here at UCLA it's $600 a person now, or how much is it? $700 now, okay, an hour. Whereas with fNIRS, we own the equipment, so when you bring a participant in, you're only having to pay them for their time. So it's the same amount of money as a behavioral study would be. It's also much more portable. As you saw in the last picture, if it's just as big as a microwave, you can put it in your car and take it to places. With the smaller unit, the NIRS Sport, that's more textbook size, and so you can put that in your luggage and take it wherever. And because it's wireless, you don't have to plug it into the wall to run it. Yeah?
I know they've only had ours a couple of years, but I was just wondering what they kind of tell you the general lifespan on this kind of equipment is.
I have no idea what the lifespan is, and I don't think they really know. Our serial number is 32. There's not that many that have been made so far. And actually, we might be one of the labs, at least for NIRS, that's running ours the hardest. And so we've had some wear on the equipment happen, and so I think we are a big test case for them from an engineering perspective. How durable is their equipment and how can they keep improving it to be durable? Here's a more detailed chart that tells you more information about it compared to like MRI and EEG. There, on the left of the table there's some variables that you might care about in terms of running a neuroimaging study, and then I tried to rank them kind of from best to worst. Some places where fNIRS is not super great, and it's one of the drawbacks of using fNIRS, is that it does not have great signal depth into the brain, whereas MRI and EEG records signals from the full brain. fNIRS can only record about 1.5 centimeters into the cortex. It's also kind of in the middle between the two in terms of spatial resolution. It's got about a centimeter of resolution. It's kind of in the middle in terms of sampling rates. It's much faster than MRI, but it's also still dependent on the bold response, which we'll talk about a little bit more in a sec. And that is inherently several seconds slower than actual brain activity, which is what EEG is measuring. The cost is kind of around the same ballpark as EEG, sometimes a little bit more expensive, sometimes less. But places where it really shines is its portability, because you can take this machine pretty much anywhere and there's few accessories that you need for running it in other places. Whereas EEG, the machine itself, is smaller than MRI, but it's pretty complicated to bring anywhere, and you also need lots of accessories like a delicate mesh cap, some electrical something gel that helps you get better signal through some people's hair and things like that. And then, of course, an MRI is bolted to the floor. Motion tolerance, fNIRS, is really great about that. You can't have the optodes moving around on someone's head, but so long as they're secured really well to someone's head, that someone can be moving their body any way they want. Whereas with EEG, you can't move muscles around the head because those give off much bigger electrical signals than the brain does. And with fMRI, you have to be laying stationary in the MRI scanner, or else you throw off the positioning that you've done with the equipment there. And then finally, you've got participant comfort, which can be a really big concern for you if you're doing studies, especially in non-standard populations. One of the reasons in the 90s that fNIRS really gained prominence was because it was an ideal neuroimaging method to use on infants that you can't really put into a big noisy MRI scanner, because there's also safety concerns with MRI. You can't have certain people being in an MRI if they've had any sort of implants that would be ripped out of their body by the magnetic field. With EEG, you're not laying down in a risky scanning environment, but you, again, can't really move during any sort of experimental paradigm with EEG, and often there's like goop on your head, which is not terrible, but kind of inconvenient, you know, especially if you have to do anything else during the day. Whereas with fNIRS, the only thing that could be considered not great is that the cap that's used to affix optodes to someone's head, it has to be snug. And so if that's pretty snug and someone's head is kind of sensitive to that tightness, that might get uncomfortable, but there's no safety risk, and you can move around and relax in whatever position you want while you're actually going through a NIRS study. Some evolution of fNIRS. It's thought of as a relatively new technology. It's based on infrared spectroscopy, which is, of course, very familiar to anyone who's a chemist. The first kind of medical use of an infrared spectrometry was from Glenn Millikan in the 1940s, when he developed this blood oxygenation device that would clip to fighter pilots' ears, because during World War II, they would be pulling so many G-forces in dogfights that they would pass out and then crash. And so they wanted a way to deliver oxygen to pilots before they passed out. And so they had this oximeter that you could clip to someone's ear, and it could detect when oxygen was getting dangerously low. From there, Franz Jobst is the first to use NIRS to measure cortical oxygenation in humans in 1977, and found it to reliably measure oxygenated hemoglobin, a known component at this point of the BOLD response. In 1988, there was some algorithmic achievements that helped us measure and quantify brain activity changes, and then from there, the first commercial system was developed, and since then you can see on the graph that probably mid-2000s it's really taken off in terms of popularity, at least in the academic realm. Now that being said, it's nowhere near fMRI is, because fNIRS is the little blue bars at the bottom and MRI is the black ones. And the equipment is still under what I would call active engineering, so they're still always improving the quality of the material that's being used to measure activity, the engineering design, how light and comfortable the headgear is, and things like that. And the field still lacks consensus on best pre-processing analysis methods. There are lots of software packages out there for analyzing data, but they all have pretty big holes in them in terms of how usable and flexible and capable they are compared to the software that MRI uses, which is a big deal when you find a little problem in it, like they did a few years ago, but that are used really extensively by a large community and so are well maintained and pretty usable. Now let's move into how fNIRS works. So briefly, I won't go into too much of the neurophysics itself because we can spend a whole class on it like Monty does in the fall here at UCLA. But really briefly, just so you kind of get a sense of it, the BOLD response stands for the blood oxygen level dependent signal. as neurons fire in your head, they consume metabolic resources, which then need replenishing. So oxygen is one of those resources, and that takes a ride on the protein called hemoglobin to the local activation sites where neurons are firing. And this relationship between the vascular dynamics and the neuronal firing is called the neurovascular coupling. Due to various processes like the increased blood flow to those areas, blood vessel dilation, etc., oxygen concentration in the blood around those activity areas initially decreases a bit and then surges up. Then, after several more seconds, tapers off and as activity slows. And this happens over the course of several seconds, where the peak of that bold response is happening probably 5 to 8 seconds after the initial activity started firing in the neurons. Both NIRS and MRI can pick up on this signal change in oxygenated hemoglobin as an indication of localized brain activity. Now, the way this works in MRI is you have this static magnetic field being driven through the NIRS or the MRI tube. The oxygen-carrying hemoglobin, which is abbreviated HBO, is diamagnetic, whereas deoxygenated hemoglobin, hemoglobin that's had that oxygen pop off, is paramagnetic. And this means that deoxygenated hemoglobin locally distorts the static magnetic field environment of the MRI scanner more than oxygenated hemoglobin does, which results in less MR signal being recorded in that area. There's a lot more about like proton spinning and tipping over, but we don't need to go into that. MR signal is thus a ratio of oxygenated to deoxygenated hemoglobin in local areas of the brain. Now, in contrast, just as they have done with different magnetic properties, oxygenated hemoglobin and deoxygenated hemoglobin have different optical properties, which is what NIRS leverages. Now, when interacting with a medium, light can either transmit through, reflect off, scatter into, or absorb into, like this little diagram helpfully explains. Different wavelengths of light can transmit through media in different amounts. So depending on the molecule size of that medium and the shape of those molecules, they may vibrate and absorb energy at certain wavelengths better than other wavelengths. So for instance, a prism transmits all wavelengths equally, so that's why you're able to see all colors of the rainbow come out. Whereas the oxygen-hydrogen bonds in ice absorbs more red, yellow, green frequencies of light than blue, and so that's why glaciers look blue to our eyes. Now, for NIRS, the infrared light transmits pretty well through skin and bone, which is why we choose infrared as the optical window of interest for us. But it differentially absorbs into oxygenated hemoglobin versus deoxygenated hemoglobin. They have what's called different absorption spectra. We can leverage this fact to detect when oxygenated hemoglobin concentration is changing. It's best to use infrared wavelengths in that window in the diagram there. That's the window that's best penetrates the head, and it's where oxygenated and deoxygenated hemoglobin are differentially absorbent. The way you can read this graph is on the left you have the absorption coefficient, which is essentially how much of this energy is being absorbed as the light passes through a medium of this type. And then along the bottom is the wavelength in nanometers. Near infrared around 700 nanometers is pretty visible to our eyes. That still looks red to us. 900 is getting larger. We can't see that anymore. But you'll see in areas that are like 750 or 850, the plots for oxygenated and deoxygenated hemoglobin are different. And so that's what we choose as wavelengths to care about when we're doing infrared spectroscopy. Our NIRS system specifically is using both 750 nanometers and 830 nanometers. You need 2 wavelengths to run, and in a moment I'll go over why that is. So whatever system you're using, they all use somewhere around those 2 numbers. It can be a little bit different depending on the machine, but it's good to know what your machine is using so that you can talk about that in methods when you're writing up your data. Now, brain media is not a perfect transmitter. It's not like glass, so projecting infrared light into the head, it's not going to pass easily through. It's going to scatter about a lot more before coming back out. So the lower right picture is a model of the most likely optical path in the adult human head that light is propagating through when you put on a standard fNIRS channel. So you measure the intensity of light that is returning to the surface compare it to the original intensity, and that enables you to infer the concentration of oxygenated and deoxygenated hemoglobin, and thus the amount of brain activity going on in the area between those sources and detectors. Now, to know how to do that comparison, it's time for some math. This is kind of the big physical law that we follow. Yes?
Quick question. So one electrode is either a detector or an emitter, right?
Yes.
So you don't have both in the same
Correct.
location. Okay.
You can build a system, and I believe some systems exist where one wire will contain both, but that's because you have like separate optical feeds going into this one head.
Right.
Yeah. And you still need space between whatever the source is and the detector is, because you need to measure something in between them.
Right.
Yeah. So the Beer-Lambert law is what basically our whole concentration calculation is based off of. It allows us to calculate the amount of light or the concentration of a medium, etc. And it essentially models incident light passing through some amount of medium at some concentration and some length, called the path length, and then the detected light coming out is a part of the equation too. Let's see, the molar absorption coefficient is that epsilon value, and that is just the absorption coefficient from the graph like this. And then the other important variables, again, are concentration of the substance in the media and the path length. Sometimes you'll see this equation abbreviated with no C or no L, and that's just because they're assuming a path length of 1 or a concentration of 100%. This little portion on the left side of the equation is called optical density, and that's just important to know because in the fNIRS world it talks a lot about optical density. That's the first step in computing your concentrations in a lot of the software packages, etc. But since brain tissue isn't perfectly transmitting, we have to modify this equation to account for the scattering going on. So instead you're calculating what's called the attenuation, and that is the same equation except you have, instead of just the normal straight shortest path length L, you're now multiplying the equation by something called the differential path length factor. And that just accounts for the fact that when light particles are scattering about, their path length is going to get longer, right, from this sort of random walk path that it's taking through the medium instead of traveling just straight through. And then you have to add on a g at the end, which is called the geometry dependent factor. And this is accounting for the amount of light that's lost, not due to the absorption, but to its scattering out of the system, essentially. So by adding g, we're acknowledging that more light is lost than before due to both absorption and scattering, not just absorption. And then by multiplying with the differential path length factor, we acknowledge the random walk nature of light photons in a scattering medium. Now, we don't know what g is, so we need to create a differential version of this equation to cancel g out so that we can actually calculate our concentrations of the chromophores we care about. Chromophore is a word for that oxygenated, deoxygenated hemoglobin. So we turn our equations into this, which allows us to calculate the attenuation from detected intensity values of 2 different light walks, which means we need to make two different light walks, and that is, in the way we do it, by transmitting 2 different light wavelengths into the head. That would be lambda 1 and lambda 2 there in the equation. And then the light intensity at T1 and T2 both matter, and T2 would be whatever this moment is, and then T1 is typically regarded as some baseline value. In a lot of the software packages that I've worked with, that's kind of an average light intensity over the entire course of the run. It doesn't really matter what you use as your baseline value because we're not caring about the absolute concentration values that we're calculating from this. We're caring about changes in changes of concentration. And so what it's changing from doesn't matter so much. It just might be important to know how your software is calculating it. Now finally, we can rearrange this equation and find out the concentrations of our chromophores. And again, we're not finding absolute concentrations with this equation. We can only find change in concentration, and that change would be from that baseline value I talked about. So it's this math that your code is doing under the hood. Now the pros of using this differential modified Beer-Lambert law is that we can calculate both oxygenated and deoxygenated hemoglobin separately, whereas MRI can't do that. And yet there are some downsides of using it to know about, which includes it does not give us the ability to calculate absolute concentration, like just mentioned, and it assumes a few things that aren't necessarily true. It assumes we know what the differential path length factor is. It assumes that G is constant and that the absorption of light across a medium is homogeneous across that entire length. And none of these are actually true in a real world situation. That's kind of the toy model that we assume. And we're using what's called a continuous wave fNIRS system. That is where when you're essentially transmitting light continuously and collecting light continuously. When you use a system like this, you can't measure any of those factors absolutely. So you have to make assumptions about what they are, and that tends to be not a valid assumption to make, which is not a killer to our measurement. It just introduces more measurement noise across subjects and makes it so that you can't compare activation between brain areas. Similar way that you can't really compare activation between areas in MRI for different reasons, but it's also just something you shouldn't be doing. Now there's a type of infrared spectroscopy called frequency domain fNIRS that can measure real DPFNG, and the way it does this is essentially by instead of continuously transmitting the same wavelength of light from a source into a detector, it's varying the frequency of the light it's putting out. And so it's kind of passing through lots of different wavelengths at once. And because you have all those different wavelengths and all the different ways that the light will slightly differently travel through the brain because of those wavelengths, then you can actually calculate the differential path length factor and the geometry dependent factor. But that's a way more expensive machine to get. It's not really practical, it takes a lot longer to run, and just very few of those really exist commercially. Most people who've done it have built their own, and so it's not really practical for our purposes to use. It's more of a thing that people keep developing when they're focused on, you know, the physics of fNIRS instead of using fNIRS for something else like we are. Now that we know about all this physics, we can explain why fNIRS gets its certain usability properties. In terms of cost and portability, generating light with lasers or LED that fNIRS does is much smaller, lighter, and cheaper to do than a large magnet that is required for MRI. In terms of motion tolerance, so long as the optode, again, doesn't move around on the scalp, the whole measurement system is localized to just that distance between the source and the detector. So anything else that's happening outside of that system, like the participant moving around, is not going to matter. The sampling rate is, it takes measurements as fast as you can. Essentially, you're flashing through all your different light sources. If you were to flash all your light sources on at once, a detector wouldn't really know where that light is coming from, so you have to go through your sources one at a time. And so that's going to be the limiting factor on your acquisition speed in fNIRS. So if you have a small kind of subset of channels positioned over one area, you can go through those really, really fast. If you're doing a full brain system, you have to go like that. So that'll take up some time, and it'll make your acquisition speed a little bit slower. That being said, you can do fancy things like, I'm just going to assume that light from over here can't reach over here, so I could maybe fire these both at once. Or if you're doing a dyadic study where you've split your channels between two people, those can be firing at the same time. In terms of the spatial resolution, when you're determining, it's determined by G, which we can estimate as like a general constant from prior frequency domain fNIRS work. Again, it's not going to be the exact number correct for every participant that you're running, but we kind of have like we know what this is supposed to be, and that's what enables us to move on. And so we can, from this measurement, we know that the amount of scattering going through someone's head, and thus the spatial resolution, how accurate we can be about where activity is happening, is about 1 centimeter. We can make this better by doing what's called multi-channel measurement, and that's where you have the standard 3 centimeter channel spacing. That'll get about 1.5 centimeters into the cortex. You can make another one that's a little bit longer or a little bit shorter. If it's a little bit longer, the signal penetration will be a little bit deeper. A little bit shorter, it'll be a little bit shallower. And based on comparing those two signals, you can get a little bit more accurate in terms of the location of certain activity. But that, again, requires a much denser system and is very time-consuming and requires some special equipment to do it. For short distance channels, you have to have very special detector heads that don't get oversaturated with the amount of light they're receiving. And then for signal depth, this is restricted by the channel spacing. A wider channel gets signal to bounce deeper, but with that farther path length it has to travel, you have time for more of it to be absorbed and more of it to scatter into the netherworld. So you get less signal coming back out of the brain for you to detect, and eventually you get a wide enough channel that you're not getting anything usable back, and that really decreases your signal to noise ratio. So a standard channel size is about 3 centimeter separation. And then finally, for the participant comfort, optodes must be touching the scalp for all of this system to work, and that can potentially be too tight, but that's the only risk that a participant will experience.